7 minutes, but I'm not quite sure that I... So other than all the other speakers, we are at 20% of this project, as Rebecca told you this morning, so there's very little data I can show you. So this is about an animal disease, and the idea is that with New Zealand's dependency on income through agriculture, that it might be appropriate to have a vaccine in the program that addresses a veterinary disease. And we have looked at a few, and the one that is very high on the list of farmers is bovine viral diarrhea. It's a disease that causes quite significant production losses, probably talking about $150 million a year that the industry loses, and another $40 million to keep track on it. Before Mycoplasma bovis came into the country, it was actually some discussion around eradicating it, and eradication would be a vaccinate, test, and slaughter combination. That's currently off the table with the costs we had from Mycoplasma bovis, but there's still interest in New Zealand and globally in a vaccine. The current vaccines are all attenuated virus or killed virus, and they're not working very well. So that's the idea. The virus is a pestivirus, single-stranded RNA, two types. Type 1 is more common and relatively mild, and type 2 is more severe and kills more animals. New Zealand has a lot more of type 1. But it's hurting productivity. And there are subtypes, and the nasty thing about it is that if it infects an animal at a short period of about a week, 10 days during pregnancy, then it produces what's called a persistently infected animal. So that animal does not show any signs, but it spreads the virus like crazy in the herd for the rest of its life, and you don't recognize them. And the other idea why we liked BVD is that there's a lot of similarities to coronavirus. We have a spike protein analog, which is the E2 protein that sits on the outside of the molecule, of the virus. It's reasonably well characterized, and unfortunately it is not very well conserved. So that's one of the molecules that the virus very quickly mutates to escape immunity. But we have a good strain that we think we can use the sequence of, and Rebecca's working on making us RNA from it, and we hope that we can get that to work rather quickly in a cattle model. We will test two variants. One is the E2 protein by itself, and then the other one has a signal peptide attached to it that comes from bovine R6 to make sure that we get a bit of secretion of the protein. And then we'll use the platform to its full strength. All the thinking around the RNA modifications and capping and LNP is all coming out of the platform. So for now, we're not worrying about any of this. We will have to later, and I'll come back to that. We will then, hopefully, in a few weeks, test expression in a bovine cell line that we have. We're currently making sure it's BVD free because it comes from the US, and most of the bovine cell lines that come from the US are actually BVD infected. But we have alternatives, so if it is, then we can work around that. And then one of the beauties with a veterinary vaccine is that we don't have to do preclinical trials. We go straight into our animals. A calf costs $80. It's not much more expensive than a mouse, and you don't have to have cages and people feeding them. You just put them out on the pasture and be done with it. So that's what we plan to do in January and February. We will look at one or two commercial vaccines as a comparison. They're both failing in New Zealand mostly because they're not against the strain, that BVD strain that we have here, but they give some immunity, and they're the golden standard that we have, or the silver or bronze standard that we have. And then we will test our two RNA vaccines, E2 and E2 plus the R6 signal peptide. And we have the dosing and everything figured out based on what we know about vaccines. AgResearch has quite a long history of developing vaccines. We used to have our own production facilities, but the government, in its wisdom, decided we need to sell them in 2004. So we have to restart things, but thankfully we have people like the platform now that can help us. And then the readouts are straightforward: antibody responses via ELISA, ELISpot for interferon gamma, which is another thing I should mention. Cattle are not mice and humans. We don't have a catalog of 600 pages with monoclonal antibodies against everything. We maybe have a page full of antibodies against bovine proteins. But we do have all the reagents for interferon gamma, because that's our standard test for bovine tuberculosis, which we are the national reference lab for. So we can do that. And we are, let me check them all. We are reasonably up to speed, and we hope to have something to actually show about April next year. And then the fun starts for me. This is a study that is really to show that we can do an RNA vaccine in livestock. There's one registered livestock vaccine in the US for pigs, but otherwise there's none, and there's a lot of, I think, unreasonable, but the questioning around does it work in cattle and sheep the same way. And another fun part of this is that I'm now getting about 1 to 2 emails a week from people that tell me, I want my cattle RNA free, so please don't do RNA vaccinations in cattle. And I refrain from explaining to them that an RNA-free cattle wouldn't really work, and that their standard steak contains about 18 to 20 grams of RNA. So that's just the fun side of it. What we want to do is we want once we have this proof of concept, we want to look at a broad coverage RNA design, and the hope is that against about 100 strains that we find in New Zealand with different sequences for the E2, we can develop an artificial sequence that hits most of these variants. That's the number one. The number two is we really need to work on the cost of goods. This is cattle and not people. Our market analysis has told us our cost of goods per dose is at about $1.80 US. So we have a long way to go to get there. What I've heard earlier today was quite encouraging, actually, to see all the effort that goes into upscaling and speeding up the process and making it cheaper, but it's still a long way to go. The other problem is ultra cold chain is not an option for agriculture, really. You need something that's lyophilized and that can sit in the shed for a week without going bust. But again, what we've seen today gives me some hope. We have the opportunity to upscale. William Ralston's facilities could do that for us, and he has all the expertise for the agricultural application. And then, yes, the first aim is to do something that works in New Zealand, where the commercial vaccines fail, but there's also an enormous global market, and there's a new aspect to animal health now, and that is greenhouse gases. If animals are healthier, you need fewer of them, and they produce less greenhouse gases. And there's new papers out there that say you can reach up to 20% of greenhouse gas reduction through healthier animals. And vaccines are something that farmers understand and use and like to use. So if we can help with that, then we tick that box as well. And the last thing is registration of the vaccine in the veterinary space is comparably straightforward. In New Zealand, it's regulated through AZVM, that's Agricultural Compounds and Veterinary Medicines. That's a group that sits within MPI. It takes about 4 to 5 years, 6 years, to get a vaccine through the process. You need to show safety, you need to show residuals, meaning that none of your vaccine is left in what people eat, and you need efficiency, which is least important for them. And you can do that in 3 animal studies with a rather limited number of animals. And if you do it right, then you can cover most of what you need for a US registration as well at the same time. And I have a team too. That's the Animal Health Solutions team in Palmerston North. And we never have the time to get everybody on one picture. So Mallory, who is here and is doing most of the work, is sitting on the shelf. But also a huge thank you to Sarah and Rebecca that have been really, really cool to work with to develop this. Thank you.